Okay, good afternoon to everybody again. I just want to briefly introduce the NABIAM project. The main goal of the NABIAM project is to establish a long-term cooperation network of contacts and relationships with relevant entities in the field of research, development, and business, in the field of polymer electronics and medical technologies, with a focus on nanotechnology, biosensors, and additive manufacturing, which basically reflects to the shortcut of the project. This network, combined with the mapping of competencies in the above mentioned fields, should help in the future to quickly create project consortia for specific topics and adequately fund them through appropriate funding programs at regional, national, and European level. So the goal is basically to bring research institutions and also companies performing research closer together for future cooperations. We are 3 partners involved in the project. The coordinator is Profactor, which is a non-university research company for applied production research located in Steyr in Upper Austria. Profactor research topics are focused on micro and nano fabrication using nano imprint lithography, additive manufacturing using inkjet printing, and also combinations of those 2 processes. Partner on the Czech side is the University of South Bohemia in České Budějovice. It's science and research center specializing in education and research in natural, human, and social sciences. In this project, it's represented by the Institute of Applied Informatics and Institute of Physics and Biophysics and Institute of Chemistry and Biochemistry. Business Upper Austria is Austrian partner. It's a business agency of the Upper Austrian government. BizUp is an innovation driver and partner for location, development, and settlement of companies, and also establishes cooperations and provides advisory services in public funding. BizUp is represented by the plastic cluster and the medical technology cluster, which are part of the BizUp. So what are activities of NABIAM? Unfortunately, I have to say that about the activities I have to already talk in the past tense, basically, because this is the last month of NABIAM. The project ends with end of September, and during the course of the project, we organized reciprocal scientific lectures with focused on the project topics, nanotechnology, biosensors, and additive manufacturing. We organized also online matchmaking event where we paired partners from companies and research institutions and companies to companies, of course. And we are also preparing a competence map, including infrastructure, equipment, and available technologies. And we are also organizing webinars where we are presenting competencies which we acquired during this competence mapping. We already had 2 webinars on nanotechnology and biosensors. And as I said, we also prepare future projects, so follow-up project of NABIAM within European calls like Interreg, Horizon Europe, mRNet, and others. And we are constantly looking for partners. So if you are interested, don't hesitate to contact us. If you want to be part of the competence mapping, your company or your institution, you can also participate in the survey, which is conducted by the NABIAM project, where you can fill if you are active in the field of nanotechnologies, biosensors, or additive manufacturing, plus some details about what you are actually doing and which infrastructure do you have. You can also follow activities of NABIAM on social media. We have LinkedIn and Facebook channel, and of course web pages. Yeah, then that's basically all from my side. And today we have 6 very interesting speakers. The first is Professor Jaroslav Jacak from Fachhochschule Linz, and he is giving us a talk about additive manufacturing in medicine. So now I'm stopping my presentation and giving my word over to Professor Jacak.
Hello. Can you hear me?
Yes, we can hear you well.
Super. So I'm I'm going to talk about additive manufacturing medicine, but I will focus on some special topics that is very of a huge interest in our group. It's in vitro modeling or mimicking of vessels, and I will give you an idea how we do this, and I will talk about in vitro model of human blood vessels wall, especially in a blood-brain barrier. So the idea is that we have a arteria, we have particles which are transported, and inside of a blood flow in this arteria. And what we would like to observe and analyze is the transport of these particles across the membrane or across the cell layers.
I'm sorry for interruption, but you are not
Yes?
sharing your
I'm
screen
sorry.
currently.
Maybe now. Do you see it?
Yes.
Perfect.
I just want to mention that the time slot for speakers are 20 minutes. I will also measure time, so sorry for interrupting if you are too long.
No problem.
And
I will manage.
to the audience, there will be a Q&A session after the presentation. Raise your hand, or you can raise your hand, and I will give the word over to you after the presentation. Thank you.
Thanks. So again, we have a flow here in the arteria. We have different cell layers, and we would like to mimic it, and we would like to observe transport of particles, for example, high density lipoproteins, low density lipoproteins, extracellular vesicle viruses across this membrane. How do we proceed? What was the idea? So we would like to make it all under flow conditions. So we designed the microfluidic chip, and this microfluidic chip needs to be co-cultured. So we would like to have more than one cell type inside of the microfluidic. That's why we decided to choose 2 microfluidic parts, let's say a dual channel microfluidic. So we have a flow on the top and flow in the bottom, and it's separated by a membrane. And in order to have a direct contact between these 2 cell types, which are cultured in the upper and in the lower channel, we have a 3D structure in the middle, or 2 or 3D, we can choose it basically, in order to have the cells interacting with each other. And this whole design of the microfluidic is suited for fluorescence microscopy, and especially for single molecule fluorescence microscopy. In this case, we can monitor single transport events across the cell layer. Yeah. So how do we print the structure that is basically allowing the 2 cells to interact with each other? Because when we just make the hole, the cells will basically grow into the chambers, and it's not what we want. So we use 2-photon or multi-photon lithography in order to write this structure. And the idea of multi-photon lithography is more or less simple. You use a femtosecond laser, femtosecond pulsed laser. You focus into a resin, and in the focus of the resin, you get sufficient intensity in order to cross-link your polymer. Of course, it's a radical polymerization. You need photoinitiators, and basically by moving your objective, you are allowed or enabled to print 3D structures. And we typically use not stereolithography with 1 photon absorption, but 2 photon absorption. In this case, we have this 2 photon absorption allows us basically to only excite our photoinitiator in a small volume inside of the focus, and this is a nonlinear process, which means the localized excitation is proportional to the second of the irradiance. Good. This with this, our system is a custom made by Workshop of Photonics. We have designed our own workshop of photonics, build it up for us. We have basically two wavelengths that we typically operate is a 515 nanometers femtosecond pulsed laser and at 1030 nanometers a femtosecond pulsed laser, so it's frequency doubled laser. We have pulse duration of around 300 femtoseconds, and repetition rate is 1 megahertz. And when we focus them into our resins, this is what we can achieve. The feature sizes are around 200 nanometer or even 100 nanometers. And when we write 3D, structures of around several micrometers can be structured. And when you look at it closer, here you see nicely the point spread function, which has been basically polymerized into the volume of the monomer. And in the past, we worked a lot with functional polymers, especially during my time at the JKU. I was working with different types of resins, and we were able to combine them into one single structure. So we used polymers with different reactive groups on surfaces. For example, here, and this is a structure consisting of 3 different types of polymers. The one is polyethylene glycol based polymer, which is protein anti-adsorptive. Then we have a green one, the green lines there. Of course, they are not green in reality, but we have just painted them green. In green means NH2 reactive, and red means thiolene reactive. So we have 3 different, let's say, types of polymers mixed together. And with using some techniques, additional techniques like stat lithography, we were able to push this feature size and the resolution of lithography down to 50 nanometers and 120 nanometers. So and further, we have also developed several techniques for immobilization of proteins. For example, in this with this structure here consisting of 2 polymers, we were able to immobilize proteins on top of the bluish volumes, and the rest was inert against protein binding. And the third thing that we have developed is photografting. So we use photochemistry in order to paint molecules on top of our surfaces. For example, here what you see is a BSA structured BSA passivated surface, and on BSA we used fluorescently labeled molecules, and we bleached the fluorophores, and due to the radical let's say forming due to photo bleaching, the fluorophores interacted with the surface and were immobilized exactly on where we wanted them, exactly where the focus beam was. So in total, we have developed a toolbox for micro to nano technology with functionalization for polymers. And we use this in our approach. So in order to bring these 2 cells directly into contact, we have taken this membrane. First, we use laser cutting. We cut out certain shapes. In this case, it was a star. Then we were writing to photon, let's say a polymeric carrier structure on the border in order to make sure that um, that we get rid of the, let's say, not nicely cut plastic leftovers, and then we printed a 3D structure into this hole. The whole membrane thickness was 63 micrometers. The material of the membrane was PET, so it was easily to cut, and we were basically due to 2 photon structuring, we were able to structure in the ranges of micrometers down to 100 nanometers. So this is what we have done basically. So we were able to also vary the stiffness of the polymer. So we had hard lines and softer lines. How we determine it? We determine it using nano-indentation via atomic force microscopy, or we also were mixing photoresist having functional and non-functional uh, uh, polymers, which means for us functional in this case were polymers which were capable of protein binding, and the red ones were inert. We used we used let's say some way of multiplexing, so we were able to write several of these structures next to each other. So all this allowed were co-cultured, all these structures they were on one membrane and cut out and written and afterwards co-cultured with two different cell types. So when we look closer into the structures, we see nicely that this is a 3D features. We have a different periodicity on top. Then we have connectors here and a different periodicity in below. Why is it so on top? We wanted the cells simply to grow on top of this grid, and the second cell should enter the holes, and so they were the holes on the back were bigger, and so they were able to enter and interact with the other cells. We were always at the beginning we were trying not to write the 3D structures, just 2D structures, but what we have experienced that the cells really like it when it's 3D, so we had more cells crawling into these holes and interacting with the upper layer of cells when there was a um, 3D structure in between. And we also varied the geometry. Here you nicely see the one area we filled up with sub elements, and the sub elements were in the range of 100 nanometer hanging. Then we were playing with the photo polymers. We were checking the biocompatibility. First we use human umbilical vein endothelial cells for biocompatibility testing. The cells, as you can see, these are the nuclei of the cells growing on top of the structures, were nicely covering everything. On control photoresist, the density of the cells was lower. They were not they were doing fine. They were not dying faster, but they were not growing as fast as on the biocompatible polymer. In our case, biocompatible polymer was a metacrylate based type of polymer which was quite hard. It was in the megapascal elasticity range in this case. Good. So now to the microfluidic, this is how it looks like. We had this first channel, second channel, then we had the objective here for illumination. The building of this channel was quite a challenge because the working distance of our objective is only 320 micrometers, which means this is the distance between the lens and the focus in this case. With respect to the glass slide, so it was all together. So we needed the lower channel especially to be very flat, and these were the two inlets and the microfluidic, and here you see the membrane, and this was of course the structure. And the most important part about the structure was not only biocompatibility and a certain amount of stability to withstand the cell growth, because the cells also are affecting the structure by simply pushing it during the growth phases, but also transparency when it comes to microscopy. So that was an important issue. If you look at the technical details, this is about the height of the channels here and the flows that we were able to achieve. And in this case, I have to mention that we only had flow on top, which was continuous. In the bottom, the cells did not require it, because if you imagine, this was more or less the part of the blood vessel. So there were the cells that are like, for example, HUVECs, which are in the first contact, let's say, to the flow. And in the other channel, in the inner channel, these were the cells which basically either muscle-like cells or parasites when it comes to blood-brain barrier, which basically stabilize the endothelial cell layer. So here was the flow was not required. This is like the innermost part of our arteria, and in this case here we only needed to change the medium in order to keep the cells alive. Good. So this is the idea. We have flow here, we have flow here, first cell layer, the second cell layer, and then the cells grew here in between, and the cells from the bottom went inside and interacted with the cells on top. So when we what was very important, and we used primary HUVEC cells, and we used also endothelial cells which mimic the blood-brain barrier, is when the cells are exposed to a flow, they expose also a polarization in the cell. Polarization means there is a difference between top of the cell, which is pointing towards the flow, and the bottom of the cell pointing towards the rest of the cell culture, or to the tissue. And what we have seen that we nicely see that there is apical and the basal part, and that the cells basically have a, a polarization on top of the cells, on top of the inside the microfluidic. Sorry. Yeah, so basically apical and the basal part has been formed. You see nicely, this is a plot which describes the stains. So CD34 was here, CD31 was here, and this is a profile of a cell membrane. So this is the lower membrane, this is the upper membrane, this is the nuclei. So which means you see, if you look at this, should be the middle of the cell, the Z position. You see that CD31 was more expressed on top, and CD34 was more expressed in the bottom of the cell, which it basically introduces a polarization. So this is how it looks like, our structure that we demonstrated the co-culture, which means we had a first layer of cells, this which has been seeded. Then we waited for 3 hours just to let the cells land on top of this. You see the different periodicities. Here is the low periodicity, here is the high periodicity. This is where the cells can enter. Here the cells cannot pass through. You nicely see the red, blue colored cells are the cells on top, and they form nice junctions. The red colors are the tight junctions, which means they're very closely connected, these endothelial cells, which means they basically form a barrier. Yeah, and the green ones are parasites. We stained the RNA. Yeah, and we incubated for several hours. Unfortunately, they do not survive this staining for a long time because RNA is blocked, and so some processes are not so efficient and they don't feel well. But for imaging, it was fine. And you see that these are the parts of cells that you can see, and they came from the bottom. So you see here cells. Of course, these are also the nuclei of the cells where the RNA abundance is highest, that came from the bottom and interact with the top layer. The top layer, in order to get it so tight, it is not an amount of a matter of hours, it's a matter of days. So they were cultivated under flow conditions for 5 days prior to the adding of the second cell type. And now what we have done, as I mentioned, we wanted to look at single molecules. So we wanted to observe the transport processes at a single molecule level. And what we nicely were able to show, that we can perform localization microscopy. This is a technique which enables to localize single emitters in 3D with a precision of let's say tens of nanometers range, so 50 nanometers lateral localization and 80 nanometers axial localization. And the thing is, what you see here is it was on a fixed cell, so we had to kill the cells, let's say, and fix them and label the cytoskeleton. And what you see here, this uh, let's say homogeneous, more or less, area, not too many stress fibers, is the area, or is basically the volume of the cytoskeleton of the endothelial cells on top. And we also looked at Hdl transport. So what you see here, these are single Hdl labeled fluorescently labeled low density, high density lipoprotein particles, which have been added from in the top channel, and they basically have been taken up by the cells. And what you see now in this video is single particles, which are basically moving inside of the cells, which have been taken up and are diffusing around. So when you look at uh, look, we can look at the trajectories. I'm not going to show you the 3D trajectories, but what you nicely see here, color coded, is the Z position, and what you see here is basically the volume that they are traveling in within a cell. So this is for example, during the observation, this was the volume that one of the particle was traveling uh, in the cell. So to sum up, what I was showing you is basically how to a possibility to mimic a barrier, and we used for this 3D printing inside of a microfluidic biocompatible photoresist, which allowed fluorescence microscopy, and we designed a chip for high NA imaging, which allows basically single molecule imaging on the chip. And last slide, so the most contribution to this work was by Boris Buchroth and Sandra Maya, and thanks to my group and of course all the people who and the institution who are involved and sponsored this project. And thanks to you for the attention.
Thank you very much for a very interesting presentation. Are there any questions from audience? Just raise your hand. I would have one in the meantime. You mentioned, as I understood from your presentation, the detection method is basically fluorescence.
Yes.
So is there a room for kind of sensor integration like an electronic circuit to detect the number of molecules or something like that?
The number of molecules for tier measurements, of course, there is a possibility. I think we haven't tried, but what would be interesting to bring on electrodes on the membrane and in the lower channel in order to also measure the let's say the resistance of the cells, and this would be a nice measure for density of the cells, which would be quite fast. And what we have also performed is electrophysiological measurements, but it's not in this particular paper, but in general. So you could also measure currents across the cells, ionic currents, if sensors in this direction would be, of course, interesting.
Yeah, I was also thinking about kind of impedance sensor, maybe also kind of resonance mass sensitive sensors to detect the amount of molecules. Yeah, interesting.
Yes, of course. What is interesting is also the what we were been showing is the uptake of the cells, but there is also lots of particles which are passing across the membrane, let's say across the cell layer into the lower channel from the top channel, and detection of those one might also be interesting.
Very nice work. Thank you.
Thanks.
Are there any questions from the audience? If not, let me introduce the next speaker. It's my colleague Michael Kainz from Profactor, and he's going to introduce our project on implants, and he's also presenting inkjet printable formulations for biocompatible materials. So, Michael, the floor is yours.
Thank you, Pavel. Can you hear me?
Yes, we can hear you. Your presentation is currently in the not in the presentation mode.
Yeah, so. I know.
Oh, perfect now. Yeah.
Yeah, now it is. Everything is good. Yeah. Thanks, Pavel. So good afternoon, everyone. Thank you, Pavel, for organizing this event and for also giving me the opportunity to present a current project that we're working on. And I'm really excited to talk a little bit about additive manufacturing of biomaterials, especially when used in an inkjet printing process. So the project that we're working on started this year. It's called InkPlant, and it's short for ink-based multi-material fabrication of next generation implants. And recent advances have actually enabled 3D printing of biocompatible materials, as shown in the presentation also before, and which may lead in the future to engineer fully functional tissue and to basically address the need for transplantation of certain tissues. And 3D printing of biomaterials have been around for quite some time, and it, several research groups have actually addressed the challenges for skin tissue engineering, cardiovascular grafts, and cartilage regeneration, just to name a few of them. And within our InkPlant project, we are mostly addressing the challenge of bone tissue engineering by using additive manufacturing techniques. And one goal of the project is to develop materials for an inkjet printing process and to basically create tailor-made biological constructs with desired physical and biological properties. So what our project is actually all about. So when you look at the left-hand side of the slide, so the vision of our project is to use additive manufacturing to fabricate patient-specific implants for four different use cases. For osteochondral defects, for the reconstruction of a meniscus, for the cleft palate, and also subperiosteal implants for the upper and lower jaw. And I would like to explain the concept of what we're trying to do in a more detailed way by using the subperiosteal implant as an example. So the sketch that you can see on the right-hand side is a rather simplified, but I think illustrative picture of what we're trying to do. So current dental implants are screwed into the bone. And the long-term predictability of such implants is directly associated with the quality and also the quantity of available bone. And after tooth loss, underlying bone is consecutively resorbed, and this is due to the lack of the mechanical stimulation. And then very often an insufficient amount of bone is available for conventional implantological techniques. So this is why we propose the following design for such a problem. So we propose that an implant to tackle this challenge should consist of two different phases, also manufactured by two different additive manufacturing techniques. So we call it basically that the implant consists of two phases, so the hard phase and the soft phase. And the hard phase should provide the right mechanical performance of bone tissue and consists of two different ceramics. For example, this could be an interconnected lattice structure of hydroxyapatite and zirconia. And on top of this ceramic part, we aim to use inkjet printing to fabricate a biomimetic soft phase, so a biomimetic coating, so to say. And for this biomimetic coating, we aim to use three different materials. So one can now ask the question, why three different materials? So it is very, or it seems unlikely that any single material will have all the properties required to mimic certain tissue functions. And when you think about it, almost all human tissue consists of either complex gradients of one material or even different materials. And each material has a specific biological and mechanical influence. And we propose that this bioactive coating should consist of three different materials, which is basically taking the hydroxyapatite in a nanodispersed phase, a peptide hydrogel, and as well as a biodegradable polymer to provide sufficient mechanical stability on top of this hard phase. So, as I mentioned, the hard phase basically is produced by biomaterial lithographic ceramic manufacturing process by our project partner Litos. At Profactor, we are developing the process for multi-material inkjet printing of the composite phase, and combining these two approaches should basically enhance the osseointegration by really mimicking bone tissue with this multi-material approach. So when thinking about the different biomaterials, so the term biomaterial is a very broad term, so to say. So in the project, we want to cover a very broad range of biomaterials, so ranging from calcium phosphate ceramics provided by the company Litos to either soft materials like hydrogels and synthetic photopolymers. So, as I already explained from the slide before, the hard phase is provided by the company Litos, which could be basically an interconnected lattice structure made out of the ceramics. And the inkjet printed soft phase consists of the Nano Hydroxyapatite, the hydrogel. So the Nano Hydroxyapatite, we're working very closely together with the company Fluidinova. The hydrogel, we're developing it together with the company 3D Matrix. The biopolymer is developed at the Johannes Kepler University. And we also try to reconstruct the meniscus by using inkjet printable PDMS, which we are developing closely together with the company Elcam and Statusys. So how do we actually develop an ink? And I would like to show you some progress that we did since the project started. And I would like to explain this by showing you the approach for the nanohydroxyapatite ink. So basically, to mimic bone tissue, one needs to first understand the natural constituents of bone. And from the literature, it is basically known that bone is a nanocomposite material consisting of hydroxyapatite nanocrystals embedded in a collagen matrix. So basically, bone consists in its macroscopic constituents out of collagen, hydroxyapatite, and water. And we are using rod-like nanoparticles to formulate ink formulations because hydroxyapatite appears as rod-like or plate-like crystals in bone. And there are a certain range of commercially available nanohydroxyapatite pastes on the market, which have basically shown or which are reported to be biocompatible, and you can use them for osteoconductive bone substitute materials. So the development process of an ink formulation starts by first identifying certain additives that can be used. For example, if we talk about photopolymers, we need certain monomers, cross-linkers, stabilizers, and photoinitiators. When it comes to developing nanoparticle-laden inks, you need like a dispersant to prevent the nanoparticles from aggregating within your ink, certain surfactants to tune the surface tension to the desired range, certain viscosity modifiers, and humectants to tune in the viscosity. So if this stage is more or less completed, one can start by formulating first inks. And when you think about an inkjet printing process, there are basically 3 to 4 major challenges that need to be overcome. So inkjet printing is very demanding when it comes to the rheological properties of the ink. So there's a very small window for viscosity in order to be able to use the ink in an inkjet printing process. So we're talking about 8 to 13, 8 to 20 millipascal seconds, and we can go up to about 60 to 80 degrees Celsius centigrade printing temperature. And when you think about 8 millipascal seconds, we are basically in the same order of magnitude as water. So very, very liquid inks. Surface tension should be about 28 to 35 millinewton per meter in order for the ink to wet within the nozzle plate of the printhead and also to wet on the substrate. When we're talking about an ink that contains nanoparticles, one should be able to control agglomerations or to hold agglomerations below one micrometer. And as there are acting very high shear stresses in the nozzle plate or on the nozzle plate, usually Newtonian fluids are used. So I tried to basically illustrate how much time it takes for every step in an inkjet printing process development. So basically identifying additives and formulation of inks takes the longest. If we have like first formulations, we are trying them basically out on a lab scale printer. We're developing curing strategies, and then we're going for industrial printheads. And while doing so while having printable ink formulations in parallel, we can start with biodegradation studies, in vitro studies, mechanical testing, and also long-term stability testing of ink formulations. So I made a small video from a LabJet or lab scale printer so that you can basically imagine how the jetting behavior looks like on a printhead. So the nozzle diameter when we're talking about inkjet printing is about 20 to 21 micrometers, and the droplets that you see ejecting from the nozzle plate, they have approximately a volume of about 10 to 20 picoliter. So I tried to illustrate the importance of being able to control the agglomeration behavior in an inkjet printing process, especially for our nanohydroxyapatite ink. So basically, as I mentioned before, we're talking about nozzle diameter sizes of about 21 micrometers. So it's really, really important to prevent nanoparticles from agglomerating prior to printing, because agglomerations can basically clog the printhead, and usually this is an irreversible process. So when one nozzle is clogged, basically the printhead is damaged. And yeah, we tried first printing tests and tried to formulate first formulations. So basically we just checked the jetting behavior, deposition of material, and try to print like thin layers, as we want to use this as a bioactive coating, so to say. The idea of using hydroxyapatite as a bioactive coating is not really new, if you could imagine. So the idea has been around since the 1980s, where research groups tried to deposit hydroxyapatite coatings on titanium implants, for example, by using airbrush or spraying methods. But being able to use it in an inkjet printing process, one can really control the deposition of material on the substrate, trying to print material gradients, for example. And yeah, I'm really happy that we can use the nanohydroxyapatite in an inkjet printing process, because I haven't seen this before. And just to get an idea of how such a printhead looks like that we are using, So this is an industrial printhead, and such a printhead consists basically of 2 to 4 nozzle rows, and each printhead has about, let's say, 380 to 1,300 nozzles. Usually they have a printing width of about 5 to 7 centimeters. So now I showed you basically how is the development process of an ink. And within the project, we're also building our own prototype printer, because obviously there is not a single system on the market that can process 3 to 4 different inkjet printable formulations. So we made an initial design of the printer so far, and I'm just going over through some components that are really crucial for our process to work. So usually inkjet printers consists of a holder where you can stack several printheads. So as I told you before, we are going to print photopolymers and water-based inks and also hydrogel, and all these materials have specific demands on the printhead. So we identified one printhead for one specific material. We have several curing possibilities on the printer, so ranging from UV LEDs to an NRR lamp. And so for the inkjet printing process to work, not only a printer is required, but also an ink supply system where you basically fill in the ink and the ink gets transported to the printhead, and some controllers and electronics to make it work. So now usually patient-specific implants are not flat, but rather curved in one direction. So it's really important that we are able to print on curved substrates. And this can be done by following a 5-axis printer approach. So the printer we are currently building, or the printer prototype, consists of three linear axes, so X, Y, Z basically, and 2 rotational axes, which are marked here as the dotted lines, so that we are able to control the rotation of the implant under the printheads, and to be able to print on basically concave substrates. I've also included some pictures of where we are now, so that you can basically see it in real time. So here's a short screenshot of the holder with the different printheads that we are going to use. With some electronics, the UV lamp is already mounted. And yeah, we hope within the project we can then process the 3 to 4 different materials to engineer this biomimetic soft face on the ceramic part. And yeah, I'm now at the end of our project, of the presentation, and I just want to thank all the partners involved, because this project would not be possible without so much support from all around Europe. So basically, I've now focused more on the materials supplier part, but there are also partners ranging from medical university to IPPE from JKU to Biomed Center from Germany, and also the partners who doing like the in vitro tests from the Charité in Berlin, from the LBI in Vienna, and also from the Trans Tissue Company and the BTI, who really support us along our journey. So thanks for your attention, and I'm happy to take questions.
Thank you, Michael, for your presentation. Are there any questions from the audience? Raise your hand, please, or write in the chat also. It's also possible. In the meantime,
Very nice presentation, Michael.
Thanks.
In the meantime, I can maybe ask on the sedimentation, because you presented a lot of inks with nanoparticles, crystals, and so on. So is sedimentation an issue for such inks for you, or how do you handle with this in the future?
Sedimentation is actually a huge issue from our side. So usually for the inks we are developing, we are doing long-term stability studies. So we're storing the inks at elevated temperatures, say 40 degrees Celsius, for a period of 30 days. And we can check the sedimentation behavior of the inks, because usually sedimentation behavior comes hand in hand with agglomeration behavior, because you can imagine particles agglomerating do sediment with a larger velocity and not staying stable in solution. So yeah, this is a really big issue that needs to be controlled in order to engineer a stable ink that can be used over several months.
I suppose that in the industrial system, using the industrial head, you will use also kind of recirculation, right?
Sure. Yeah. This is then
the solution mixed.
This is when you use it in the process, but when you store the ink without using it, sedimentation is actually a big issue. Yeah.
Okay, thank you. I don't see any hand raised. So let's continue with the next presentation.